Hi everybody. In this section of my short lecture, I will talk about early fundamental findings in sensory neuroscience. So these are few landmark discoveries or findings in neuroscience that advanced the neuroscience a lot. One early significant finding is the discovery of the neural cells called the neurons. Before the discovery of the neurons, scientists used to believe that in the brain, huge amount of fibers are connected to each other in a very complex manner. However, they didn't think the presence of the cells, neural cells or neurons in the brain tissue or nervous tissue. They used to believe that huge presence of fibers in the brain tissue. However, first a neuroanatomist whose name is Golgi, probably you have heard this word, he discovered a stain which was named after his name, Golgi stain. And this person, a very famous neuroanatomist who is considered as a father of the neuroanatomy, he is actually the person who first identified the presence of neurons in the nervous tissue. His name is Ramon Cajal. He is a Spanish neuroanatomist. His early life is very interesting. He was not a gentle boy or child. He was kind of unruly, and his father was a professor in a medical school in Spain, Madrid, and his father was a physician as well as a neuroanatomist, and he was taken to the prison for his violent behavior once he made a cannon. And when he was around 11 years old, he was very talented but kind of unruly. He used that cannon, own invented cannon, to destroy the gate of entrance of the house of his neighbor, and that was the incident that took him to the jail. Anyway, so his father tried to control him, and he was taken to a graveyard once with his father. Since his father was a neuroanatomist, he used to go to the graveyard to dissect the dead body and see the structure of the body, and he was taken with his father, and he gradually became interested to understand the human body, and eventually he got admitted to the medical college where his father was a faculty, and he became a doctor, physician, and he also became a anatomist. He studied anatomy, and mainly he focused on the brain tissue. First, he studied the peripheral nerve tissue, then he studied the brain tissue. Anyway, so what he did, he used a lot this Golgi stain, and injected the stain in different neural tissues, and identified the neurons, and he was very good in sketching, drawing, and he created hundreds of elegant, sketches that showed how the neurons are present in the neural tissue and how they are connected to each other, and how different neurons, different types of neurons are present in the brain tissue. You see here pyramidal neuron here. These are different type. You see many branches or dendrites. This one doesn't have many. So all different kinds of, you see, neurons he uh, discovered in the neural tissue, and very nicely presented. That's why his sketches are still used in as reference in neuroscience. Okay, and in many books and many articles, you will see that his sketches are being used. Okay, so this was a great discovery by Raymond Cahill. And then if you see another interesting discovery which was made by a British scientist, Charles Sherrington, who won Nobel Prize in 1932. In 1906, he articulated what are now recognized as the fundamental principles of brain function. He explained that a single neuron cannot accomplish a function. To accomplish a function, a network is necessary. So neurons must get connected to each other to form the network, and that's how the neurons perform the function. A single neuron is not capable to perform or execute any function. So to accomplish or execute a function, neurons must form network, okay, through synapses. And he also proposed that the axon terminal of the previous neuron gets connected via synapse to the dendrite or the cell body of the next neuron. So this is a neuron, this is the axon terminal, this is the cell body, these are the dendrites, okay, and this is the next neuron, and these are the dendrites, and this is the cell body. Okay, so the axon terminal here of previous neuron forms synapses, many synapses with the following neurons or next neurons, and the axon terminal forms synapse with the dendrites. This is the dendrite of next neuron, this is axon terminal, or some axon terminals can directly form synapse with the cell body. So this is the cell body and this is the axon terminal. So axon terminals of the previous neuron form synapses with the cell body of the next neuron or the dendrites of the next neuron. Okay, so that's how the synapses are formed, and he mentioned that something happens here at the synapse, and he was able to measure that the signal, when the signal is given to the next neuron, the signal gets slow at the synapse. Okay, but he didn't know what makes the synapse signal transmission slower at the synapses. Okay, anyway, so these two are very important findings in neuroscience. One is neurons must form networks to execute a function, and how they form network by creating synapses. Okay, so Interestingly, you will learn later that uh, his findings were very important. Why? Let me just briefly talk about that. When your visual part of the brain uh, processes something that like a structure that has a concave and a round or convex part. So this is the convex part, this is the concave. This is the round or convex, right? Okay, and this is like we'll say flat. This is flat, right? So this object has four surfaces. For example, one is concave, another is convex, and two flats. So now uh, a single neuron cannot process this whole object. So for the flat surfaces you need, for example, these two neurons, and for this concave surface you need this neuron, and for convex surface you need this neuron. Okay, and all these neurons need to be connected to each other to share the information to give you the complete picture or shape of this object. Makes sense? Now we know that the different neurons in the visual part of the brain are excited by or activated by different shapes, and in an object that has multiple shapes in it, you need activation of those neurons to give you the shape of the object in the brain. Okay, so this is very powerful finding. And he also mentioned that not only that, different groups of neurons need to talk to each other. So within the network as well as between the groups of neurons, networking is needed. For example, this object, these neurons are giving the shape of this object, right? But this object might have other features. For example, texture. Is it smooth or rough, the surface? So if it has texture, these neurons cannot process this. What is the color of this object? So for example, these neurons process the color, these neurons process the texture. All these are visual neurons, right? All these are visual properties. So these group, if these are group A, these are group B processing the texture, group C processing the color. So all these groups also need to talk to each other between them. Okay, so that will give you the complete picture. Um, the synapse was also proposed by Sherrington, and as I mentioned that beautiful pictures of those synapses were presented by Ramon Cajal. He did draw all those different types of neurons and different types of synapses in the brain, how they are connected to each other. However, as I mentioned that although Sherrington, he mentioned that something happens in the synapse that slows down the signal transmission, but he did not know what is that slowing down the signal transmission. Another scientist, his name is Otto Louis. He first discovered that a chemical that is called neurotransmitter or neurochemical is released at the synapses, and that enhances or inhibits the transmission of the signal. So it is not electrical, electrochemical. That means some chemicals are released at the synapses, and that transmits the electrical signal from one neuron to the next. Okay, and he performed a simple but very elegant study where he took the frog heart in Ringer's solution to keep the heart alive for a while. So this is the Ringer solution, and he took a frog heart here. So this is the heart of the frog, and this heart is still beating or contracting and relaxing, because the Ringer solution will keep it alive. So even after you take it out from the frog and put it in this solution, the heart will continue functioning. Okay, and this heart, he just only took the heart out without the vagus nerve, which is attached to the heart, and he took another container where in the Ringer solution he placed another heart, frog heart, but this heart had vagus nerve connected to it. Vagus nerve is the autonomic parasympathetic autonomic nerve, and what he did, he stimulated this vagus nerve. Okay, electrically stimulated this vagus nerve, and after giving some electrical stimulation, then what he did, he poured this solution from this container to this container. Okay, and what he found, that after he poured this solution, Ringer solution, from here to here, this heart contractions slowed down. Okay, so the contraction of heart decreased, the number of contraction, that means heart got slower. So that clearly indicates that something was released when he stimulated the vagus nerve here. Something was released in this solution that slows down the heart, right? So definitely something was released here at the synapse where the vagus nerve is attached to the heart muscle. Okay, that is the synapse, and that is actually acetylcholine, ACH neurotransmitter, and that slows down the heart. Okay, so Otto Louis, he first discovered that neurochemical or neurotransmitter is released that slows down the transmission of the signal. That is another very interesting and powerful finding, neurotransmitter. German scientist Johannes Miller in 1826, he mentioned or proposed a theory that is called law of specific nerve energies. This is also very interesting and powerful discovery in neuroscience. It states that the kinds of perception we have depend on which neurons are activated, not on what is activating those neurons. Very true. A simple example. This is your tongue. Tongue has what kind of receptors inside the taste buds. Inside the taste buds you have the taste receptors, right? So gustatory or taste receptors, And we know that the food molecules, when you put the food molecules on your tongue, the food molecules bind with the receptors, right? And when the molecules, signal molecules or food molecules bind with the receptors, then through the nerves, signal is taken to the taste cortex or gustatory cortex. That gives you the perception of taste. Is it sweet or sour or bitter, something like that. Now, uh, it doesn't matter who activates these neurons, but which neurons or receptors are being activated. What it means that now if instead of food, if I can stimulate these receptors by other means, for example, if I put an electrode and electrically stimulate or activate these receptors, then signal will digitally go to the taste cortex, okay, and taste cortex will give you the signal of taste. So brain has no way to know who is stimulating the receptors. Receptors are located in the peripheral organs, right? And brain is sitting inside a very dark cranial cavity. So brain has no way to know who is sending the signal. Okay? Brain only knows that brain has no way to know who is stimulating the receptors. Brain only knows that who is sending the signal to it. Okay, so now instead of food, if I can stimulate these receptors by other means, electrical signal will go to the brain, and brain will just count digitally how many times the signal arrive, and if the signal activates the taste cortex, you will get the feeling of taste. If the signal activates the sound cortex or auditory cortex, you will listen the sound. If the signal activates the visual neurons in the visual cortex, you will see the color or something. Okay, so it doesn't matter who is activating these receptors or earlier neurons. The matter is which set of neurons, earlier neurons or receptors are being activated. Okay, not how. So that is what it says. Depends which neurons are activated, not on what activating those neurons. Okay, and that is another another example is like sometimes people listen the sound although there is no source of sound. So some signal from the ear goes to the auditory cortex. Now auditory cortex doesn't know how the receptors are being activated. It only knows that receptors are sending signal, and how many times, that is the frequency. Okay, so now if I can stimulate the auditory receptors here by other means, then the neurons will get the signal, electrical signal, right, and will count and give you the frequency of sound. So this is a very interesting topic of neuroscience. That brain is sitting inside the dark cavity. It doesn't see what's going on outside. It only counts digitally the number of electrical signal arriving, and which area is being activated by that, and that is giving you the perception of that sensation. Okay. Another very important finding is that how the action potential is generated, how action potential in a neuron is generated or initiated and propagated or transmitted. Okay, and these two scientists, Sir Alan Hodgkin and Sir Andrew Haxley, they created a very powerful model that is called Hodgkin Haxley model. And this model explains many important biophysical processes that take place during the transmission of electrical signal, not only in the neuron, other excitable cells of the body, for example heart muscle or myocytes. So they described this model in 1952 to explain the ionic mechanisms underlying the initiation and propagation of action potential, and they used the squid axon, which is pretty large compared to human nerve axon. So this is a good model to study. Still many scientists use this model. So it was easy for them to study, since the structure is much larger than human neuron and axon. Okay, in 1963 they received a Nobel Prize for this powerful model that they proposed. Okay, so if you see that model, he considered a number of things. First, let me just briefly draw. This is a cell membrane. You know, cell membrane is a phospholipid bilayer. So this is a cell membrane, and the ion moves from one side to the other side, right? So this is extracellular fluid, and or medium, this is intracellular medium or fluid. Okay, and here this one CM, this includes this is a phospholipid bilayer, right? This whole structure, and CM indicates the capacitance of that membrane. Okay, capacitance of the membrane, and then electrical conductances were shown by Gn here, this one. So one is voltage gated and another is leak channels. Okay, so you see here this is Gn and this is Gl, leak. Okay, so one is linear and another is non-linear. So this is called non-linear N, and this is linear. Okay, so these are two other conductances, and then he also showed here IP, the source of current. Okay, the current source. So let me write it down here. So Cm is capacitance in phospholipid bilayer, and voltage gated ion channels are indicated by Gn. Okay, voltage gated ion channels, electrical conductance. Okay, and then Gl, this is the leak channel, as I mentioned, leak channel, linear conductance. So this is voltage gated non-linear conductance, that's why N, and this is leak channel linear conductance. Okay, And another is here, E, N and EL, nonlinear and linear. And E indicates the batteries, or another way we can say that E is the source of voltage, which is the battery. So source En and El, nonlinear and linear. Source of voltage, okay, which is the battery. So these are the components, and by using this model he showed the current flow through the phospholipid bilayer. So how the ions flow through the phospholipid bilayer, that is explained by this model, this theory that he showed, and the amount of current that flows is measured by using this formula, ionic current flow, Cm, okay, then dVm divided by dt, okay, of course t is the time, and membrane potential is Vm. So this is the membrane potential. So you can see here that membrane potential you can also measure by using this formula if you know the other, or you can measure the other values, okay. So you can determine the ion flow of ion through the phospholipid bilayer, as well as you can measure or determine the membrane potential, okay, across the membrane phospholipid bilayer. So this is the model they proposed, and biophysics uses this model a lot. We'll talk about the action potential in next slide. So let's see. In resting condition, different tissues have different types of cells, and different cells have different resting membrane potential. Resting membrane potential is the difference of voltage across the membrane. So if this is like a cell, intracellular fluid and extracellular fluid that I just mentioned is separated by phospholipid bilayer, right? So which is the cell membrane, phospholipid bilayer. So the concentration of different ions are the chemicals that carry either positive or negative electrical charges, right, and cations carry positive charges and anions carry negative charges. So important cations are potassium and sodium here for membrane potential, okay. Both are cations, and important anion is the chloride, which is an anion, negative, okay. So across the cell membrane, if you see intracellular, this is inside intra, and this is outside extra, and the concentration of these anions and cations are different in intracellular and extracellular fluid, okay. And because of that difference of concentration of these chemicals, ions, always inside the cell is negative. In a neuron, usually we see inside the cell or inside the axon, the voltage is minus 70 millivolt. That means negative 70 millivolt. Okay, that means more anions are inside the cell and more cations are outside of the cell, and less cations are inside, more cations outside. Okay, and that's what you see here. This is an axon. So you can see this is the axon, and if you see inside the axon and outside of the axon, this is extracellular, and inside the axon is this is intracellular, right? So extracellular is here and intracellular is here, and if you see the concentration of ion in the inner side of the membrane, this is the membrane and this is outer side of the membrane. Okay, you can see here by different colors they showed the sodium, potassium, that means those two cations, and anions, mainly chloride. And if you put an electrode outside of the axon or cell and inside another and measure the voltage outside and inside and subtract, you'll see that inside is minus 70 millivolt compared to outside. That means much more negative inside. Okay, and that's what you see here. This is showing the difference between inside and outside, and that is the membrane potential. Okay, and normally the resting membrane potential is this value, which is in resting condition. That means this neuron is not excited, it is in non-excitable condition or status. Okay, now it is easy to measure the membrane potential, that means the difference between outside and inside. Okay, and then if you stimulate the neuron, we know that if we stimulate anywhere, either stimulate the cell body or the axon, some changes will occur. Electrical signal will travel through the membrane. So when that electrical signal travels through the membrane, some ion channels get opened, and what happens, the sodium, most of the cases sodium channels will get opened when the electrical signal will travel like this through the membrane, and sodium channels will get opened. Now sodium concentration is higher always outside than inside. Okay, so outside sodium is higher, inside is lower. So what will happen when the channels will get open? Naturally sodium will rush in because of concentration gradient. Okay, and that will cause the membrane potential move towards, from here towards positive. Make sense? If this way is positive, the membrane potential will move towards positive because sodium is positive and it is getting in. So inside will be more move towards positive. Okay, and that is called depolarization here. Okay, depolarization, moving towards positive, okay, from the negative. And in some excitable cells, the depolarization occurs slowly. That means ion channels open slowly, and then when it reaches a certain level, then more ion channels abruptly open. So this part occurs very sharp first, okay, but first part occurs slowly, and this is called the threshold. When the depolarization reaches the threshold level, then abruptly the voltage gated channels get opened, and that is the action potential that generates. Okay, so this is called the threshold here. So this is minus resting membrane potential, usually in neurons minus 70 millivolt. Okay, and you can see here that it can go all the way up to plus 30 or even more towards positive. And after that, what happens, the sodium channels get closed and potassium channels get open. Now potassium is always opposite, concentration is high inside and low outside. So when the potassium channels will get opened, since the concentration is high inside, potassium will get out from high to low, right? That's the difference of pressure gradient or concentration gradient. So potassium will get out. So if potassium gets out, that means positive is now leaving the axon or the cell body, right? So that will cause downwards movement of this potential, that means towards negative again, because positive is leaving, and this is called repolarization. Repolarization. Okay, these two together is the action potential, we say. So action potential is upwards and downwards, depolarization and repolarization, two phases. And this is the threshold. If the stimulation is weak, then what we see that the membrane potential may not reach to the threshold. So the action potential will not be generated. This is the action potential. Okay, it will get truncated and eventually diminish. So this one will not spread far, will stay in local area, only the area where the excitation was presented or stimulation was presented. But if action potential occurs, then that can shoot the signal far away. Okay, so to send the signal further, you need far away, you need the action potential. Okay, and the stimulation should be strong enough that can trigger the action potential, that can move the depolarization or membrane potential up to the threshold. Okay, so here you can see that this is outside, this is phospholipid bilayer, this is sodium channel, potassium channel, and in this part sodium is getting in, and here potassium is getting out, right? So depolarization, repolarization. Now why this is very important, action potential, because for example, your eye, inside the eye you have the retina where you have the photoreceptors, right? Sensory receptors for vision. So these are the photoreceptors, and these are actually neuron-like cells, photoreceptors. Now if the light stimulates these photoreceptors, and if the stimulation is not strong enough to create the action potential, signal will not get out from the eyeball, will not go all the way to the visual part of the brain. because it is far from the eye, you know that. So that's why it is very important that action potential is generated, and that can send the signal out and shoot the signal far away to the next area where it should be processed on the way. Okay, which is true for all sensory receptors. Action potentials must be, generated, and that should be able to travel, carry the signal towards the brain. Okay, now we'll just talk about synapse, the structure and the chemical signal transmission in a synapse. I already explained in some extent. This is the axon terminal that I mentioned before. Axon terminal, this part, and this part is called this is a membrane at the axon terminal. This is called presynaptic membrane. Okay, so this part is the axon terminal and this membrane is the presynaptic membrane. Okay, and in presynaptic or axon terminal, here you have some membranous structures filled with the neurotransmitters. These are called the vesicles. So here you can see the vesicles. Same thing here. They contain the neurotransmitters, and there is a gap. This is called the synapse, synaptic cleft. Synaptic cleft. Cleft means the gap. So this is actually a gap, and this is the postsynaptic membrane. It could be a muscle fiber or next dendrite or cell body of the next neuron, right, that I mentioned. So this part is the postsynaptic membrane. Postsynaptic membrane, and pre and postsynaptic, they are facing each other. And what happens when electrical signal travels through the axon terminal, calcium channels get opened and calcium enters into the presynaptic terminal to rupture these vesicles. And when these vesicles get ruptured, they release the neurotransmitter here, or neurochemicals in the cleft, synaptic cleft. Okay, and these neurotransmitters will bind with the receptors here. On the postsynaptic membrane, you have the receptors. So these are the receptors. Okay, and these neurotransmitter molecules will bind with the specific receptors. This must be very highly specific. So the receptors and the neurotransmitter molecules or signal molecules, they should be same type. For example, if this is ACH, acetylcholine molecules, these must be acetylcholine receptors to make the binding, perfect binding. Okay, so when that occurs, it's like key lock type. ACH molecules will bind with ACH receptors. Glutamate will bind with glutamate receptors. GABA will bind with GABA receptors. Makes sense? So when that perfect binding will occur, postsynaptic membrane channels will get opened, and normally, as I explained in last slide, that sodium gets into the cell. So sodium positive will get in, and that will cause the depolarization, initiate the depolarization. Okay, So that's how the structure of a synapse is, and how the chemicals are released, and they activate the receptors to open the postsynaptic gates or channels to cause the depolarization, which is very important, right? So let's stop here, and we'll talk about the excitation and inhibition in the synapses, and few other things in next lecture.